It's not the vaccine, it's the virus. Viral mutation rates play a pivotal role in vaccine development. Mutation rate refers to how often genetic changes occur in a virus's genome during replication. All vaccines trigger immunity, but how long it lasts depends on several factors. One of the most important ones is the rate at which a virus replicates. If a virus replicates quickly, it has a chance to produce more mutations, also known as variants. The more variants emerge, the harder it is to make a vaccine that will create lasting immunity, because the target keeps moving. If a virus is stable, that gives us a big advantage. Measles is an example of a stable virus that is unlikely to replicate. So scientists could predict that immunity would last a long time, which it does. Smallpox and polio, highly contagious viruses that were almost eradicated through vaccination, are also stable with low mutation rates. It's also difficult to measure how strong immunity is over time. Antibody count doesn't give a full picture of protection because vaccines also train B cells and T cells. When confronted by a known virus, memory B cells quickly deploy new antibodies to stop infection, while killer T cells clean up where antibodies failed, eliminating infected cells. B cells and T cells are both types of lymphocytes, which are white blood cells crucial for the immune system. They work together to defend the body against pathogens, but they have distinct roles. B cells produce antibodies that target and neutralize invaders, while T cells directly kill infected cells and help regulate the immune response. Beyond antibody production, B cells play crucial roles in infection by acting as antigen-presenting cells, producing cytokines, and shaping immune responses. They can also develop into memory cells for long-term immunity. For many diseases, experts believe antibodies are the key protective mechanism, but B and T cells may be even more important for other pathogens, including tuberculosis, malaria, and HIV. While less famous than antibodies, they play a big role in making immunity last. In general, RNA viruses like influenza, HIV, and SARS-CoV-2 have higher mutation rates than DNA viruses such as smallpox. Although there are exceptions like polio and measles, the result is one shot of the smallpox vaccine lasts a lifetime, and in over 40 years of research, it has not been possible to create a viable vaccine against the retrovirus HIV. A DNA virus is a virus that has DNA as its genetic material. It replicates using a DNA-dependent DNA polymerase and can be either double-stranded, dsDNA, or single-stranded, sDNA. DNA viruses and RNA viruses differ primarily in their genetic material. DNA viruses use DNA as their hereditary material, while RNA viruses use RNA. This difference influences their replication strategies, mutation rates, and evolutionary paths. Mutations can be classified into several types. Point mutations. A single nucleotide changes in the virus's genetic code, which may alter protein function. Insertions and deletions. Additions or losses of small segments of genetic material, leading to structural changes in viral proteins. Reassortment. This occurs when two different viruses infect the same host cell and exchange genetic segments, creating a new hybrid virus. The consequences of viral mutations can be profound. Increased transmission. Some mutations allow the virus to spread more easily between individuals, leading to more rapid outbreaks. Immune evasion. Certain mutations can help the virus escape detection by the host's immune system, making it more challenging to control the infection. Vaccine resistance. Mutations may render existing vaccines less effective or even ineffective, necessitating the development of new vaccines. How do mutation rate affects vaccine development? Antigenic drift and shift. High mutation rates can change viral surface proteins, antigens, that are the primary target of vaccines. Example: the influenza virus undergoes frequent antigenic drift, requiring yearly reformulation of the flu vaccine. In rare cases, a major change, antigenic shift, can lead to pandemics. Immune escape. A mutated virus may evade the immune response induced by previous infection or vaccination. Example: some SARS-CoV-2 variants, for example Omicron, had mutations that reduced vaccine effectiveness, necessitating updated booster shots and variant-specific vaccines. Vaccine target selection. Vaccines are ideally designed against conserved regions, parts of the virus that mutate less frequently. HIV's high mutation rate across its envelope proteins has been the major obstacle in developing an effective vaccine. Need for rapid vaccine platforms. High mutation rates favor the use of flexible platforms like mRNA vaccines, which can be updated faster than traditional ones. This was critical during the COVID-19 pandemic. Strategies used to address mutation challenges. Multivalent vaccines. Target multiple strains or antigens at once. For example, HPV vaccine. Universal vaccines. Aim to target conserved elements. The stationary targets. For example, a universal flu vaccine in development. Surveillance and rapid response. Global monitoring of variants enables timely updates to vaccines. Case study: Influenza and COVID-19. Two prominent examples of how viral mutations impact vaccine development are influenza and COVID-19. Influenza. Influenza viruses mutate rapidly, leading to seasonal variations. This necessitates annual updates to the influenza vaccine to match the circulating strains. The World Health Organization, WHO, monitors global influenza activity and recommends the composition of the seasonal flu vaccine. COVID-19. The SARS-CoV-2 virus responsible for COVID-19 has also exhibited significant mutations. Variants of concern, such as the Alpha, Beta, Gamma, and Delta variants, have shown changes in transmissibility and immune evasion. Vaccine manufacturers have had to adapt their vaccines to address these variants, and updated booster doses have been recommended to maintain immunity. The dynamic nature of viral mutations presents ongoing challenges for vaccine development and public health. However, through continuous monitoring, research, and adaptation, vaccines can be tailored to address these evolving threats. The battle against viral mutations and the quest for effective vaccines underscores the importance of global collaboration and scientific innovation in safeguarding public health. To learn more about viral mutations and vaccines, read the full article in the Informed Woman magazine. Subscribe today for trusted women's healthcare insights. Visit theinformedwoman.com to access exclusive articles and stay informed about women's health.